Welcome back to Biology A. This lesson is going to focus on the last and fourth macromolecule that we've been discussing in this half of the unit. So far we've talked about carbohydrates, we've talked about lipids, we've talked about proteins as well as enzymes. And now we're going to focus on the fourth group, and that's the nucleic acid group. It's very important to us because it makes you who you are. So if you look down here in this graphic I've got, the graphic starts with a five-sided shape. We should be able to recognize that by now because we've covered that in carbohydrates. So this happens to be a carbohydrate or a sugar. It's a five-sided sugar, so it's ribose, okay? And then I want you to think of this five-sided sugar from this point on as the house, okay? This is the house. On this five-sided sugar, you're going to see a balloon pop up. The balloon that pops up has a phosphate in its chemical structure. So the phosphate group is what the balloon is going to bind it to its neighboring house. So it goes house, balloon, and house. So let me just show you what that looks like. I'll show you in a second. And then off of the other side of the house is going to be a flag, and that's a base. So the flag is going to have nitrogens in it. So let's see what this looks like. The sugar is sticky in that it has these other parts on here, right? The phosphate balloon or group that hangs off the top is going to bind the next house or ribose sugar in the chain. So the chain is going to run between sugar and phosphate, and then sugar and phosphate, etc., etc. Off of the middle of these, like flags that hang off of a house, you'll see a base, and the base is going to be different with each of the nucleic acids we're going to talk about. There are 4. So each of these bases, whenever I say the word base, think flag. Everybody has a different flag. If your people are Italian, then you'll have an Italian flag. If you are Ohio State fans, you're going to have an OSU flag. Flags can be different. The house and the balloon stays the same. You can fly whatever flag you want. Keep that in mind. If you pick from 4. 4 or 5. All right, so here are our learning, a big idea, just one more time. We talked about carbon, hydrogen, and oxygen sort of ad nauseum. And now we've just talked about nitrogen in the flag or the base and phosphorus in the balloon that's going to bind these whole things together. So we're going to talk about how the structure of nucleic acid stores information to build and maintain an organism. There's a whole 2 units on this, so don't worry, we're only going to skim the surface. We're also going to describe the structural similarities between 2 versions of nucleic acids, DNA or deoxyribose and RNA ribose. We're going to talk about that here in just a second. All right, I'm going to turn this off for you. Now, nucleic acids store and transmit genetic information. So when I say they make you who you are, literally the reason you have blue eyes and brown hair or dark skin or big feet are all because of the sequence of nucleic acids that you have in your DNA. So they store and transmit genetic information. So here's that structure again. Now, the 5-sided sugar you see, carbons and hydrogens and oxygens. Now, in RNA, ribonucleic acid, the little foot here has an oxygen as well as a hydrogen. So this is a proper and normal ribose sugar. DNA, or deoxyribonucleic acid, is deoxy, right? There is no oxygen. So the only thing you'd see here on the foot is an H, not an O. So the main difference in structure between ribonucleic acid and deoxyribonucleic acid is that off of this particular carbon, there is either an oxygen or there's not. Why do you care? Because in the process of going from this molecular nucleic acid to an entire organism, the system that translates that information to you will go between the language of nucleic acids to ribonucleic acids to DNA, to proteins, etc. So not in this unit. Just understand that here's one main difference, okay? There are phosphate groups that bind Here is the houses again. See, here's the house with the flag. It's upside down though. That's the base. This is the roof. Here's the balloon. Binds it to the next base of this ribose house with its flag and its balloon. Here's the next ribo house with its flag and its balloon. All right, this picture is upside down. But the flags stick together, like the flags connect together if there are two strands, because the other difference between DNA and RNA is the DNA is double, D for double, stranded. So RNA would just be one of these strands. DNA is the two of them together. All right, so the two main differences. I just spoke of some of them. See if you can see it. D, double-stranded. R, ringle-stranded. You're going to have to figure that one out on your own. D for double, R for not. Okay? The structural difference is the presence of an oxygen for ribose, because that's normal. Deoxy for no oxygen in the DNA sugar. The bases that are used is critical. Okay, the bases used for nucleic acids, your options are T, C, A, or G. The difference is you get three of the four the same in ribonucleic acid, C, A, and G, but you can't have thymine in ribose. Ribose cannot have thymine, and DNA deoxyribose cannot have uracil. You don't care why, just remember that, okay? So there are three differences. 2 or double versus 1, hydrogen or with an oxygen on the other, the fact that you can have thymine for DNA and uracil for RNA. All right, and here is just another picture of it. Oops, these two are switched. Good catch. I found it and I caught it. Okay, so RNA, single, D for double DNA. Let's see, what else is coming up? If I change the bases, I want you to think about this for a second. If I change the bases, the C, T, G, A, blah, 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 if I changed that sequence, do you think it would change the protein? Well, the sequence starts where we go from DNA in your nucleus. A copy of that leaves your nucleus, and it is a copy, so it's exactly the same. And then from that copy, an amino acid and or protein is made. So this collection contributes to these 4 amino acids. This collection contributes to these 4 amino acids. So if I changed from AUG to AUC, I wouldn't get methionine anymore. I'd get something else. So I don't have them all memorized either. But if you switched out AUG for AUC and you didn't get methionine, you got something else, that's a different amino acid. And we've already learned if you change the sequence of the amino acids, you change the sequence of the proteins. So the answer is definitively yes. That's why sometimes there are mutations in DNA. We're going to do this together in class, but the differences and similarities that we've already discussed, double helix, 5-carbon sugar on both of them, phosphate group on both of them, some nitrogen base on both of them. And also if you change the sequence in any of those, you change the sequence of the protein. Single use, instructions. Oh, I should add on here while I'm here. All right, so no oxygen in deoxyribose, thymine as a base instead of uracil, oxygen in regular ribose, uracil as a base and not thymine. All right, that's it. Just want to make sure you understand the basic structure of nucleic acids and how they store information to build into proteins, describe the structural similarities and differences, and you just need to check in with yourself to see how you feel about that. I am looking forward to wrapping this unit up. That's all the content we needed to know so far, so check in with me if you have any questions. I will see you tomorrow in class.